Okay, so we do a lot of research, the Western Center. We have 3 PIs that do multiple studies over the year on agricultural settings. So whether that are environmental research on agricultural settings. And we focus primarily on water. We've been doing water quality research in the West now, I think going on probably 12 years or something like that. But before that, we were doing all types of water quality research all over the West and some parts of the US. But in 2018, we published one of the first studies of agricultural water multi-state look at indicators and pathogen prevalences in the Western US. This includes Washington State and California, the Central Valley region, and the Yakima region of Washington State. And some of the key findings were that irrigation water rarely exceeded the agricultural water standard. And all those samples were run within the hold time, and we used the standard, the method 1603, which is the gold standard in the produce safety rule. And just what Don mentioned earlier is that we figured that, or we found that indicator bacteria generally don't predict pathogen prevalence or concentration. So how much is in there? Because indicators are like this, pathogens are like this. So what we wanted to do is test some nuances of that assumption. And so we did, and we found that in general, it doesn't hold up, but it's still better than nothing. And then another surprising finding was that pathogen prevalence was, and these are pathogens that can make you sick. These are the ones that lead to outbreaks and things like that. And in particular, Salmonella, as you increase volume, Salmonella, you increase detecting a pathogen by a great deal. So this was published in German Environmental Quality, and you guys can all go look at that. If you want some mind-numbing reading, do it. Because scientific papers, that's how we roll. This is some of the data, but the one thing I want you to focus on is that the bottle to the right is generally what you collect, or what the labs collect, which is 100 mils. I think that one might be a 500 mil, but I made it tiny, super small. What we wanted to do was look at volume and what is an appropriate volume to be representative, or at least to seek out pathogens more appropriately. So what we did is we pulled that number up to 10 liters, which is equivalent to 2.5 gallons of water. And we processed those down, concentrated them, and processed those for pathogens. And what you see is that as you increase volume, you increase your ability to detect the pathogen. So and by the way, if you process 2.5 gallons of water for bacterial indicators, you're going to get a TNTC. It's just going to happen, right? So we can do these comparisons, and I'll tell you how we're helping with that, and to help growers and irrigators understand where pathogen prevalences occur and how best to sort of modify their thinking on that. So another gift from the government, and I hope this doesn't go away in the rethinking, is, and I don't think it will, because we've put a report together for the FDA to consider, is that growers can do what they call data sharing, which is section 11247 of subpart E. And what it says is that any grower on a common source, so if you look at this map here, this is a common source. So it's one irrigation district, very small in Washington State. And you've got anywhere from 15 to 20 farms there. I just made a number up, 15 farms, but there are more than that. And if every one of them has to sample, you're talking about 300 samples. And at $100 a piece, it gets pretty pricey. And you have to do it over a certain number of times. And so it does get really pricey. So what we want to do is aggregate, so basically put together all these growers as one and see where they can, if they have no reasonably identifiable source of contamination. So between you and the next person or the previous person, there's not a wastewater facility discharging into the canal, which they don't. Generally, canals are left alone. But if you're doing river sampling and you and another grower are taking water out of that river, is it appropriate if you guys just share your data? Like one guy takes a sample, and this gal is taking samples. Well, you do it 1 month, I'll do it the next. We share our data. Well, they put that in the rule that's appropriate. You can do that. It's basically ambient monitoring for agricultural water, which is really great concept. And we get this all the time. Well, will they accept that? And by they, you mean the FDA or the CDFA or anyone coming out and doing inspection. Well, it says they do not intend to limit data sharing, and that's what that's called, data sharing. As long as you meet these certain criteria, and it's written in the rule like that. So we tested that. We wanted to look at it. So we started with a baseline in 2015. That's that paper that you saw there, a baseline of pathogen, and basically we took a shotgun approach. We sent out spatially all of our water sampling sites. We sampled them over the year. We published that, and then we started to pull it down towards the grower level. So at the turnouts and running through the pipe systems and taking samples along the way to see if they lined up, if they matched up, or if they weren't statistically different. So we wanted to know, are they different or are they the same? Now, there were some challenges with that. And again, we used the data as a guide. There was some challenges with that. You see this little line here. You're like, well, that's it doesn't look like the rest, but statistically speaking, it is the same. And if you are between a source like a dairy or a confluence, does that make a difference? Well, we tested that. And I'll show you the data here on the next slide. But what we can't account for is wildlife presence. So very acute happening. So what happens at the source? So what we couldn't account for that unless you put wildlife cameras out there or something like that. And then vegetation within the canal. Are those important? Well, it's hard to test that when you can't see it. So that's one of the challenges we had. Okay, sorry, it stuttered on me. So this is approximately 25 sample sites, maybe a little more. And this is 4 years of data on these. And what we found is that if you aggregate, and that's that map I showed you, these are all the sites on that map. And if you look at this, the first half, so the first half of this data is surface water, and the second half is surface water into a pipe system that gets delivered to growers. And what we found is that there's no reasonably there was no reasonably identifiable microbiological contamination, and also that E. coli concentrations were not statistically different. So this begs the question, is what we always get. And of course, there's higher variability in the pipe systems because those came online in subsequent years. So if we sampled them as much as we sampled the surface, they should pull together, generally speaking. It's not as noisy on that side, but they're all below the 126 and 410 threshold right there. So what our thought is, well, you could take 2 samples and represent all 25 growers or 20 growers or 15 growers, and that's appropriate, right? Well, it needs to be updated always, right? So you have to have some kind of mechanism for that. But our suggestion is, yes, if your irrigation district's already taking samples, I think that it's appropriate to look at it, make sure there's none of those reasonably identifiables, but to talk to them and say, hey, I need your data, because I don't have to take a sample then. You've already done it, especially if it's the appropriate test that they're using for E. coli. Now, this was one of the ones where this was this reclamation district. We had one bad acting canal that kept popping the threshold. And so if you see it here, this is not good. So again, you're talking about a geometric mean of 186, which is way above the threshold, and 630. So again, 126 and 410 way above. But in our thinking, statistically thinking is, well, they're still not statistically different. So is it appropriate for these growers, and I think this was a group of 7 growers, could they take a sample, even though there's a shared risk profile with them? And we thought, heck, you might as well, because you all share the same risk. So, but what they can do also is, again, this is that same data, is there's another gift from the government that isn't data sharing, but it's called, I mean, it's not called, it is logged, it's a die-off coefficient that they use, not coefficient, I don't know what they call it. Anyway, there's a die-off of 0.5 log die-off per day. So if all the growers waited a single day on the same stretch of canal, they waited a single day, their numbers come down. Now that we can get into the math of all that, but if you look at the graph on the right, you see that all of them come down below the statistical threshold value and geometric mean, just waiting one day between irrigating and harvest. So they can still share that data. So these are the nuances of it that we're looking into, and what we've given to the FDA to think about on these steps. I just wanted to put this up there because it's appropriate. Is that turbidity is not your friend. So if you have turbid water, that means dirt, debris. It could be algae, green water, dirt water. It's not your friend, because it absolutely increases the odds of having some pathogen in your system. Because you can think about it. I mean, sediment underwater is generally concentrated bacteria, right? Once it's in the free floating environment, it sort of disperses. So that's why when we take a sample, and if you take a sediment sample underneath it, this one's going to be hotter. The sediment is going to be hotter than the water. So you want to keep that in mind. So if you're taking a sample and it's turbid, and you cap that and you send it to the lab, just know it's probably going to be hot. At least it has a better possibility of being hot. So you just want to either limit that source, slow down that water, however you need to pull that water, that sediment, or whatever's causing the turbidity, to pull that water down. And if your irrigation district is doing it, you can go talk to them and say, hey, it's increasing sediment. I'm popping a sample and it's getting above the threshold. So what can I do about that? Because they'll generally try and help you out. This was an old study we did looking at data over, I think, a 10-year time, and what we found in this data is that if you impound water from a well, it will generally increase E. coli concentrations. So no matter what, right? So you're taking clean water, you're impounding it, it's open to everything. So that's an obvious thing, right? I just wanted to put it out there for you guys and look at the differences between well water, which is generally low, even in the winter, spring, summer. And we attribute the rise in E. coli, very slight rise in E. coli, to draws. So people drawing water, it tends to shake up the basin. And then the reservoir, again, you get in the West, you generally get the rain in the fall and winter. So you get, and I don't know why the summer, we couldn't figure out why the summer, maybe it was just water drying down and concentrating, why those were, why that was happening, the 77 number. But if you impound water, this is the thing out of this, if you impound it, you need to generally know that it's going to increase the level. And some people use bluestone to neutralize a lot of algae. It also does a great job on, when appropriately used, on bacteria. That's copper sulfate. And I'm gonna end with a quick study we did. There was a sewage spill directly into a canal up in Yakima County. Everyone freaked out. The whole world freaked out up there. Because one, they didn't know how long it had been happening, but what sort of frightened everybody, and what most of us water quality folks know, is that utilities generally follow roads, and roads go over canals, and canals are ripe for aging infrastructure. Again, we're talking about infrastructure. And so this pipe ruptured and poured around 2 gallons, I'm sorry, a 5-gallon bucket every minute into the canal. So we got together with some researchers at FDA and said, hey, let's because they do this for oyster areas, oyster growing areas, where you could have a sewage discharge out into a bay or somewhere where they're doing oyster testing or oyster collections and growing areas. And they use this dye to act what they call male-specific colifies. They're usually looking at viruses, but they take this dye and they let it just go at a certain concentration, like it was a wastewater facility, and see how far it goes out and how long it disperses. Well, we're like, let's do that in a canal. This is fun. So, so we did. So this is the exact bridge that that the spill occurred on, and we concentrated. This is what the dye looks like when it hits the water, and it surprised us. We were like, hey, we should take a sample. This is the first thing we recommended. Take a sample right at the spill site. Well, if you do, you're going to miss it, because you see how it concentrates into the middle of the canal during the dye plume, and it runs along the side. It doesn't disperse until about a quarter mile, half mile out. And so it was really cool to see that, because it definitely changed our recommendation for accidental spills in canals. And we wrote that report to the Washington Water Resources Association, and also to the FDA, to our partners at the FDA, to sort of consider is that irrigation canals or growers should be aware of if your district has a spill plan in place. And so this is all the dorky data, the engineering data from it, but we released for 12 hours, and it turned the whole canal red for probably 2 days. But what we saw was, in all the stuff aside here, all the numbers aside, is that it's basically like a freight train coming through. You put the gates down, you let it pass through, and then it's gone. That's what happened here. Is the dye didn't linger at all. You could see a very sharp rise, and then it starts to come down, and then it's gone. And these are fluorometers out 40 miles. It was a 90-mile canal. Again, further downstream. This one was 1 mile downstream, and this is further down. What you see is that the concentration drops. Of course, you have some dilution, but it's the same effect. The car sort of widens. So you add more cars to it. Sorry about the train analogy, but it's appropriate. The car sort of widens out. You get less concentration, but it leaves very quickly and trails off down to zero, down to almost non-detect for these things, which we geeked out on. And that's my story.
All right. Well, thank you so much, both Don and Ronnie. Maybe Ronnie, if you could go back to non-share.
Yes, ma'am.
So we can get a picture of everybody. We've had a couple of comments come through the chat, but Ronnie, there was a couple of comments that I just wanted to make sure we had a little bit of dialogue clarification on. So the sodium thiosulfate tablets, or whatever they are, just wanted to make sure that it was clear. They are really important for neutralizing chlorine. And of course, if you have residual chlorine in your sample, that's going to kill off the bacteria. And so what they're used for is for water sampling, especially if you're sampling a chlorination source. So they would be really important to include if you were treating your water with chlorine, or in some cases, other antimicrobials. If you're taking water from a reservoir or canal where there's no expectation of chlorine to be present, then those tablets are are less important. So Ronnie, I just wanted to make that clarification that, you know, when you were talking about, well, it didn't matter because the type of sampling you were talking about, it didn't matter. So I'll pause there in case you want to say something.
Yeah, no, that, and you're right. So I should have clarified that. Yeah, when you're sampling drinking water out of your, especially a municipal source, it's chlorinated. Well, generally chlorinated. Yeah, you want to lose that. But a lot of people will say this to me, dude, I took a sample and that darn thing dropped into the water. You know, while they're taking a canal sample, and I'm like, stop worrying about it.
Right,
You're
right.
doing surface water sampling. Yeah. So we get that. I get that all the time. Like,
Yeah,
don't worry about
I
that.
just wanted to
But it
make
is
sure
important.
that
Yeah.
people made that connection, that the reason it's there is to neutralize the chlorines. Otherwise, you would have this really die off, even faster die off than Ronnie showed over the transportation time.
Yep.
The second clarification I wanted to make, you talked about sampling from stagnant water, which, you know, the stagnant water sources, which is kind of, we would have to just, you know, clarify what stagnant water meant. But as you were describing it in more detail, I would put a twist on that, you wouldn't sample stagnant water because that water source you're unlikely to use as agricultural water. And so when we're talking about sampling here, we're talking about sampling not for fun or research purposes. It's for getting data on the water you want to use as agricultural water, which is water that is contacting the harvestable portion of the crop. So I would just, again, I just wanted to clarify that the sampling that you're going to be doing on the farm already, you've gone through the decision-making process of, I'm going to use this water or that water. Now you might be taking samples from water that you're unsure of that you're going to use or not, and use the data to make that decision. But I just wanted to clarify that point. You described the truck running through the water, and I'm thinking, I don't know
that's completely that comment. That one, I added that a long time ago in our workshops. That's the one thing because people generally, some folks go a little bit nuts on sampling. Like, I need to get in the field, take samples. So you're like, no, water source. Stick to the water source as close to where you're taking it. Yeah.
So I mean, you can do other testing for curiosity's sake.
I do.
But what you really need to focus on is that water that you're going to use as agricultural water. And then I wanted to clarify on the laboratory output and the TNTC. So there, on that report, there was both coliform counts and E. coli counts. But for the purposes of testing for, you know, agricultural water quality metrics, we're focused on the E. coli, right? And so it's really important that you get a count for the E. coli. But I think it's less important that you get a count for the total coliform. Am I correct there?
Right.
Okay.
Yes.
Okay.
Yes, you're correct. So the reason I put that in there, one, you can't find many like images that have a report, and I could have put my municipal one up. Municipal one, it's perfectly appropriate for a TNT, not a TNTC, for a less than number, which is generally how they report those, less than, because the test doesn't go below 1 or below 0 or at 0. But the TNTC one is generally, for E. coli, you need that number. And if they give you TNTC one, you need to resample anyway, but you also, for the next sample, I guess I should clarify, and I'm glad you're doing that, is that for the next one you say, guys, I got a TNTC, you're going to need to do something with this to give me a number, because I need to know how far I need to come down. So, anyway.
Right.
Yeah, they'll give you a
So yeah, I just wanted to make that clarification.
Awesome.
So The one other, we did have a comment in the chat, and I'll encourage anybody else who's on the line still with us here to put in comments in the chat or questions in the chat. Dora asked if there was a basic guide or a checklist for growers surrounding testing, and so Dawn provided a link which we'll make sure is on our website if it's not already there, a decision tree about sampling. But we also have 2 videos that feature Ronnie on aseptic sampling and preparing to sample, and they're short and really to the point. You know, I highly recommend you having a look at those videos as a potential for training. Zan just indicated that the link is already there. So, and another question in the last webinar on the first webinar actually on the water series, there was an assessment for growers for a risk assessment, a checklist for assessing water quality risk. And the question is there a similar tool in Spanish? And I don't think there currently is, Ronnie.
We do. Yeah, our assessment tool, I think we converted to Spanish. So the not Western Center, WIP's question I'll get them and I'll post it to you. I'll give it to you, Linda, and you can discuss.
That would be great. Perfect. Okay. Yeah. Okay. So if you have other languages other than Spanish as well.
Yeah, I think we have Spanish and I want to say Vietnamese. I can't remember the others, but we have a couple.
Okay. Perfect. That'd be great. Thank you for that question. So we will make sure that we have those up there. So, Dawn, do you have any questions? Ronnie, do you have any questions?
From my standpoint, I just wanted to share that in 10 years of GAPS/regulatory education efforts, it turns out that very few people fully understand where those numbers come from and what they mean. So I hope that it was useful to go through some of that, and I appreciated the chance to go through some of that so that we have an understanding of both what they mean and how to get reliable numbers to tie it into what Ronnie presented today. I think the no data is the only, bad data is our enemy. We can't make we cannot make good decisions based on bad data.
Yeah, I mean, I knew that, you know, the numbers came from, were old and had evolved over time. But I didn't know the entire history. So I really appreciate that.
Well, yeah, and Don said it, and it should be said, is that those epidemiology studies were exhaustive, and they sort of complain about FDA taking those recreational standards and applying them to FSMA. But if you look at it, there are no epidemiology studies done on any other type of water like these that are appropriate to make these decisions. So they took the best available science and implemented it into the rule, which was good. I think I mean, it's effective. It's what we got.
Yeah, it's a good starting point. I mean, you have to draw a line somewhere.
Yeah, right.
And soon we will see what version B of that is. So, okay, we have another question. What is the best practice if we have several wells pumping into one pipe or one reservoir? Should I test all my wells or just the main source? So either one or both of you can answer that question. And
well, it depends. Well, here's the scientific answer, and Don says it depends. So are you using that water in the reservoir specifically, or are you piping that water to a field? And so that's where you'll want to That's where your your system inspection comes along. You're like, well, I'm only using it, I'm piping it this way, but I'm putting it in the reservoir and using it later. Or maybe not, you're using it at all, it's just a backup. So if it's just a backup, nope, you don't need to test the reservoir. If you're using that water, yep, you need to test the reservoir. So wherever you draw that water,
Right.
and out to the field.
I think you want to make sure that you, again, that whole picture and definition of what is my agricultural water on my property. So if agricultural water is the water that's touching the produce, then you're viewing it from that standpoint of where is the water that I'm actually applying to my crop? Where is that coming from? And if it's not coming from the well directly, or from a well through a pipe and then, out, but it's always going to that reservoir, you're just using those wells to fill a reservoir, and then you're drawing the water that you're using in your field from that reservoir, then it makes sense to sample the one place, which is the mixing part in the reservoir. Yeah.
And if I can just add on to that, though, by sampling just the wells, one piece that you miss is the vulnerability where the water is open in the reservoir. If you have wildlife or domesticated animals that you know about or don't know about that got access to it, you would only capture that if you measured from the reservoir. That said, if your reservoir is showing problematically high numbers, the first thing that you would probably want to do in terms of problem solving is test each of the wells to find out if it's a source problem or if it's a holding problem.
Right. That's a really good point, that there might be times when you would want to have data on the wells. And there might be a situation where, you're mostly using the reservoir, but maybe there's 2 wells on your, farm, a subset of wells on your farm that you also use in direct application, and you might want to have 2 data sets, right? Because currently the standards for testing open water sources is different from testing well water sources. And so I think it gets back to that, again, very beginning, defining what your ag water is, doing a risk assessment of your system, trying to reduce risks that you have some control over, so that you're starting from a point as positive as you can make it, and then sampling in a way that, and I use the word representative because that's really what you're trying. The goal here is that you understand your water quality from a microbial standpoint. And so the sampling techniques, the where you sample from, how you sample, is all kind of geared towards you having a reasonable chance of getting a real number. And that goes to how you store the samples and how many hours it takes to get to the lab. All of that is for you to get an honest, best as you can do representation of that water source. And we all acknowledge that if you have a reservoir and you took 100 samples from that reservoir, they're not all going to be identical. But as Ronnie showed with some of his data, you're going to get a range, but statistically, it's going to give you over time a representation of what that water quality is. Yeah, and that's another reason of why there's more than one sample required currently.
There you go. That's what I was just about to say.
Right.
That 126 is on 5 samples, typically 5 and 30. So it's always multiple samples, not a single sample. That single sample maximum has been eliminated. So yeah, so it's always over time how many samples. And if you take them all in one day, you're not going to get a representative sample. If you take them spread out over a month, you're going to get a representative sample. That 126 is appropriate. So Yeah, that should be said. That's a good point, Linda.
Right. And then the idea about the as currently written, it's getting this profile as close to harvest as feasible. It depends on what kind of farm you have and what kind of growing. If you're harvesting over a 6-month period, then the harvest period for that water is 6 months. If you're harvesting over a 1-month period, then it's a little, or you're growing over a 1-month period versus growing over a 6-month or 12-month period. So it changes depending on what kind of operation you have, what kind of product you're growing, how that water is being used. And so as Ronnie pointed out, that season can have an impact on that quality. And from a food safety standpoint, the most important part of that quality is again, the stuff that's most likely to contact the harvestable portion of the crop. So it all kind of ties together.
I can just comment. One of the things that you brought up, Linda, was our goal is risk reduction, and understanding the quality of the water is one step. But as you said, the definition of agricultural water isn't just about knowing the definition. The less the water contacts the produce, the lower the risk. So understanding the water and making decisions, including, well, maybe if our application method can be changed operationally to reduce the amount that the water contacts the harvestable produce. I mean, that's a great risk reduction step. Just to reinforce that idea. Sometimes people don't have choices about the water they use, but you do have a choice about how you use the water.
Mm-hmm.
I like to use an example of tree crops, for that. So you can often structure your irrigation water for a tree fruit or a tree nut such that the irrigation water is not going to be in contact with the harvestable portion of the crop. So you can, kind of take off the table irrigation water. And so then you're looking at water that's used for crop protection sprays. And it might be possible that for the limited amount of water that's needed for crop protection sprays, that you would be able to use a higher value source water, direct from groundwater or municipal water for that narrow application, or within a month of harvest use a more restricted water source for that narrow application. So that's part of that kind of whole thought process and approach to risk reduction on the farm, which comes with that annual assessment of your risk. And as I said, at the beginning of this program, we realized that the growers in California are, having water this year is a very elevated concern, and may alter where people are actually getting their water from, and may require some real-time evaluation of water quality, especially if it's a change over water sources that have been used in the past to one that is more risky generally than perhaps one has been using in the past. So those are considerations to take. It might also, result in greater use of drip or greater use of low water application methods that might actually end up reducing risk. So, all right. Well, I've really enjoyed the conversation today. Thank you, everybody, for attending and for joining us over the last few sessions. And